Good morning, everyone. I'm Adriparna, and I'll be talking about the origin of life, disproving the RNA World Hypothesis. As a biology student, I was always intrigued by the science behind the origin of life, and being so actually helped me to analyze the theories and the concepts in much more depth than any other people, and hence I'm here with the topic. So, as I said, for a very long time, it's been the quest of human beings to find out the origin of life. We did trace our origin to single-celled organisms, but then we were stuck. We thought that we might find the answer in our genome, which is the DNA, but there arose the problem of the egg and the chicken, which says that the DNA stores information. We know this, that the DNA stores information which forms the protein, but on the other hand, the proteins catalyze the reactions which form the DNA instead. So, which one came first? That's the problem here. Whether the DNA first formed the proteins or the proteins first catalyzed reactions to form the DNA. There came to our rescue RNA, which could do both. In layman's language, this is what RNA World Hypothesis is. That's what started the origin of life, according to this hypothesis. From the early contributors, Orgel, Crick, and Woods, we come to know that RNA stored both genetic information and catalyzed the chemical reactions in primitive cells. But in later evolutionary time, DNA did take over as genetic material, but the transition from the RNA world to the DNA world was never complete because there are very fundamental reactions to date which are catalyzed by RNA itself. So it's very aptly said that RNA are the molecular fossils of an earlier world. Now, points of the RNA world hypothesis say that Earth's chemistry was forming random grains of RNA, and RNA could replicate, evolve, interact with its surrounding environment, as well as compete with each other. This was, of course, possible because of base pairing, because if an RNA strand is kept in a medium surrounded by various, adequate number of nucleotides, then the nucleotides will base pair up complementarily with the bases in the template RNA, thus forming a complementary replicated RNA strand. That's how it could replicate itself, and any kind of mutation in the replicated RNA could eventually lead to natural selection and thus cause evolution. We know that RNA is very reactive and unstable, owing to the fact that it is a single helix structure, so it's quite exposed to the outer surroundings, and thus it's very reactive to the chemicals present in the outer surroundings. The second carbon of the ribose sugar, of course, consists of the hydroxyl group, which makes it quite reactive again, and the uracil makes it quite unstable. Thus, we see that RNA is able to function as all these different things, like replication, evolution, interaction, as well as competition. It can do all of these things because of the fact that RNA is very reactive and unstable. So what we come to know is that RNA is to be an all-rounder according to this hypothesis. RNA could actually self-replicate with the help of ribozymes, which in itself is an RNA. And it could also catalyze reactions to form proteins. Eventually, with the change of function and chemistry, RNA started coding for DNA, and eventually DNA became the master template, and thus what we see now is that the DNA forms RNA and eventually the proteins. The relics of RNA world hypothesis, like the discovery of ribozyme by Seck, Altman, and Gilbert, actually made the RNA world hypothesis believable, because ribozyme is an RNA which functions as such an important catalyst in human beings. And also we know that some of the viruses have RNA as their genetic material, which proves that RNA actually still functions as a catalyst as well as a genetic material. Secondly, in acetyl-CoA, which is such an important component of a Krebs cycle, we can see this nucleotide, which is also present in RNA, which suggests that this acetyl-CoA actually evolved from RNA. Also, similarly, in vitamin B12, we can see the same nucleotide, which is this, and we know that this nucleotide is also present in RNA, thus proving that vitamin B12 as well as acetyl-CoA actually evolved from RNA actually. Next comes splicing, which is such an important phenomenon in eukaryotes. In this spliceosome, which contains more protein than RNA, spliceosome actually consists most of it of proteins and less amount of the spliceosome is actually RNA, but the crunch here is that it uses RNA as the molecular scissor to cut the introns and join the exons, right? So RNA is actually the reaction center in spliceosome and not the protein, which suggests so strongly that RNA was such an important component, and it still is. Peptidyl transferase is again derived from RNA, which helps to catalyze the reaction of joining the adjacent amino acids to form the primary structure of the protein. But here arises the question, which is, how did nature form the backbone of replicated RNA nucleotides? In the initial part of my presentation, I told you that if we keep an RNA in suitable environment, then the nucleotides present there can actually combine as base pairs to the template RNA. But it's still not known that how the nucleotides, the adjacent replicated nucleotides, could combine with each other. Therefore, we don't know that how the phosphodiester links of the replicated nucleotides could have formed. Of course, these are things which can be done in present-day laboratories, but we don't know how this could have happened in the wild when there was no RNA polymerase present, no ligase present, which are actually proteinaceous. How could this have happened in the wild is not yet known. And then again, we don't know what precedes the RNA World Hypothesis, because RNA is too complex a molecule to have arisen prebiotically. How could all of a sudden such a complex molecule come together to function so differently and diversely? Then again, the most important thing is the concept of proteins first, which I'm going to explain now, because many scientists have proposed this concept, but many still don't believe this. But before that, points to be noted are that it's been shown in the labs. It's been shown in the labs that if ribose sugar bases and phosphate groups are present in the primitive earth, then they could spontaneously join together to form the nucleotides. But because bases in the RNA chain are highly reactive and critical, nucleic acid formation wouldn't have been possible spontaneously. Though nucleotides could have formed, but nucleic acid formation couldn't have been absolutely possible without the aid of proteins. Now, RNA replicated nucleotides can be joined in the lab, as I said in the previous slide, but how in the wild is not yet known. I'm not saying that not known doesn't mean it couldn't have happened, but if we can find an alternative solution to these questions, then definitely that should be considered. Because we know that Urey and Miller experiment and later experiments by Bada proved by simulating conditions of primitive Earth, amino acids were formed actually. So there are two things. First of all, amino acid formation and nucleic acid formation, which happened first. We can see that nucleotides could not have combined spontaneously to form the nucleic acid. But on the other hand, we can see from the Urey and Miller experiment that primitive Earth could have formed amino acids, which proves that formation of proteins were easier than formation of nucleic acids. Thus, I hypothesize that amino acids actually formed before nucleic acids formed, and amino acids actually joined together to form proteins. And after that, simple proteins catalyze the reaction to form the nucleic acids by forming the phosphodiester bonds of adjacent nucleotides. The second point actually clears our question in the previous slide of the bonding of phosphodiester linkages, the replicated nucleotides, right? Simple proteins could actually form the phosphodiester bonds, and nucleotides were evolved. Thus, nucleic acids took up the job of serving as the building blocks of genetic and transmission material as we know them today. I clearly state that amino acids were formed previously to nucleic acids, and proteins were formed, which eventually led to the catalysis and formation of nucleic acids, and thus the present-day nucleic acids were formed, which is RNA and DNA, eventually taking up the job of the RNA World Hypothesis. So basically, proteins first came, and the protein world was created, and after that, the RNA World Hypothesis can be believable. Thank you so much. It was my presentation. This is my bibliography. Thank you for listening.